Hi and welcome to IGCSE Biology. In this video, we are covering enzymes, one of the most important topics in your entire exam. Before we start, I want to show you an exam question right here on screen. Explain why only one type of substrate can bind to a specific enzyme. By the end of this video, you will know exactly how to answer this question using the precise words the examiner's mark scheme look for. So let's get started. So what exactly is an enzyme? First, let's understand the word catalyst. A catalyst is any substance that speeds up a chemical reaction without being used up itself. The last part is important. It is not consumed. It does its job and remains unchanged. Enzymes are biological catalysts. They are proteins. And this is something the examiner tests. Proteins, not carbohydrates, not lipids. They are proteins in nature. Their job is to speed up the metabolic reactions happening inside living organisms. What are metabolic reactions? Metabolic reaction is every chemical reaction happening inside your body right now. From digesting food to building proteins and releasing energy. All of these are metabolic reactions. Without enzymes, these reactions would happen far too slowly to keep you alive. So look at the key facts on the right hand side of the slide. These are the examiner tested points. Enzymes are proteins. They are not used up. They control metabolic reactions. Without them, life cannot be sustained. For example, amylase enzyme breaks down starch. Protease enzyme breaks down proteins. Catalase breaks down hydrogen peroxide and lipase breaks down lipids. We will come back to these short after some time. So, how do we name enzymes? There is an easy rule. Most enzymes name ends with ASE, ASE, and the name of enzyme tells you what they break down. Now look at this table. There are three columns. First column, enzyme name. Second column, substrate. Substrate means the substance on which the enzyme will work on. Third one is product. Product means what is formed after the reaction. So amylase works on starch and the product is maltose. Maltase acts on maltose and the product will be glucose. Sucrase will act on sucrose and the product will be glucose. Protease enzyme acts on protein and the product is amino acids. Lipase enzyme works on lipid and it gives products fatty acids and glycerol. Catalase enzyme works on hydrogen peroxide and the products are water and oxygen. This table is examiner gold. Learn these names, substrates and products because they can come up directly in your papers. Now enzyme specificity. This is a favorite exam concept. Each enzyme can only work on one specific substrate. Means amylase enzyme will not act on protein. Protease enzyme will not act on starch. It means that the enzyme is specific for its substrate. Only one specific substrate can attach onto the specific active site of the enzyme. You have to remember these keywords, substrate and product. Substrate is a substance on which the enzyme works on, and the product is a substance that is formed after the reaction. Now the most important part, how does an enzyme actually work? Look at this diagram. Every enzyme molecule has a specific dent in it called the active site. This active site has a very particular shape. Then this substrate is the molecule the enzyme is going to act on and it has a specific shape that fits perfectly onto the active site of the enzyme, not just roughly, perfectly. We say the shapes are complementary to each other, so that's why they are getting fit into each other. So when the substrate slides into the active site, they join together to form what we call an enzyme-substrate complex. This is temporary. It only lasts for a very short period of time. Inside this complex, the enzyme causes the substrate to change. It either breaks it into smaller molecules or joins it into another molecule. So the result is called the product. After the end of the reaction, the product releases from the active site. And what about the enzyme? Enzyme will remain completely unchanged, unconsumed and free at the end of reaction, and it will be free to do other reactions and take up the next substrate molecule. So you have to remember 4 keywords, 4 key terms that can appear in your exam. Active site, complementary shape, enzyme-substrate complex and the product. Now, we have to stop here and look carefully because this is the most important slide in the whole video. Look at what students write in the exam, and this mistake happens constantly. When asked by an examiner that why an enzyme stops working at high temperature, students write the enzyme was killed by the heat or the enzyme died. Wrong, completely wrong, and the examiner will give you 0 marks for that. Let me explain you why. Enzymes are not alive. They are proteins. They are biomolecules which are protein in nature and they cannot be killed or die. What actually happens is, at high temperature, the active energy increases so much that the enzyme molecule begins to change its shape. The active site changes shape. So when the active site will change its shape, the substrate can no longer fit into it and they will no longer remain complementary to each other. So there will be no enzyme substrate complex forming and no product is forming. So basically the enzyme has stopped working. This is called as denaturation. The enzyme is denatured. This is the word you must use. Not killed, not dead. Denatured. Same rule applies for extreme pH. Because if the enzyme is denatured, it will not perform its function. And enzymes perform their function at the optimum temperature and pH conditions. If the temperature gets too high or there is an extreme pH, so enzyme functioning will be disturbed, will be stopped, and we call it that enzyme is denatured, not killed. So write this down and note this term, denatured. Now you must remember this question from the start of the video. Here it is again. Explain why only one type of substrate can fit into a specific enzyme. Now you can answer it. Let's build the perfect answer together. First point. Each enzyme has an active site with a specific shape. Second point. Only a substrate with a complementary shape can fit into that active site. That word complementary is key. Use this. Third point is when the substrate binds, they form an enzyme-substrate complex. Fourth point is other substrates have a different shape, so they cannot fit. Only one type of substrate works perfectly for each enzyme. So look at these keywords at the bottom of the slide. Active site, complementary shape, enzyme-substrate complex and specific. If you write these words in your answer, you will get the marks. If you leave them out, even if you understand them, you will not. So you have to use these keywords. Before we finish, let's log these 3 things into your memory. Number 1. Enzymes are proteins. They are biological catalysts. They speed up metabolic reactions and they are not used up. Number 2. enzyme has an active site with a specific shape. The substrate has a complementary shape that fits perfectly into the active site of the enzyme. This forms an enzyme-substrate complex. That is why each enzyme only works on one substrate. Number 3. And this is a mark winner. Enzymes are not killed, they are denatured, the active site changes shape and the substrate can no longer fit. That's it for the video 1. In the next video, we will go deeper. How exactly does temperature affect enzyme activity? How does pH affect it? And what happens at the molecular level during denaturation? If this helped you, please subscribe. See you in the next